We've discussed the concept of mutations. We've shown how this triple mutant binds. I now need to get to the point of this section of our discussion of what does it mean. The concern to all of us is, if you have this mutation, does it mean that antibodies are no longer effective? Does it mean that convalescent serum is no longer effective? Does it mean that you could get in reinfection? And even of greatest concern, does it mean that the vaccines won't prevent us from getting infected by this mutation? I'm pleased to say that Dr. Tulio de Oliveira today has broken some news on this subject, and I will ask him to present this subject matter in a lecture presentation to us so we can hear directly from him his findings on the ineffectiveness of convalescent serum. Tulio, please go ahead.
Just some simple information. One of the main reasons why in South Africa, both myself and my colleagues, we look very careful at mutations is because for the past 20 years, we have been looking at mutations on HIV and TB, tuberculosis, that normally associated with resistance to treatment, what we call multi-drug resistance pathogens. So we got well trained on infectious diseases of looking for mutations and try to understand how they work. Yeah, we have been working basic day and night in South Africa and many colleagues around the world during all the holiday season to try to understand more how these mutations interact, but also how the body cell defense would be effective or not against this virus. So this presentation is just highlighting a little bit the emergence and rapid spread of a new SARS CoV-2 lineage or variant with multiple spike mutations in South Africa. Yeah, and then just to start, it's always good to highlight the tool that we are using. One of the main tools is called Genomics. What it means is to generate genomes of the virus. Yeah, and that has been such a potent tool in COVID-19 fight. It was used to discover the virus that caused the disease, yeah, which was the SARS CoV-2. It was, and we did that, and the scientific community did that in weeks, yeah. And then was used to develop diagnostics, yeah. Was also used to develop vaccines. Was used to track transmission, to identify reinfection, to understand virus and host interaction, and lately to identify new variants that transmit very fast. So genomics has been one of the cornerstones of this response to the pandemic, which over 350,000 genomes produced, with 3.5 of those in South Africa. Yeah. So today, as I speak to you, yeah, just came. We have been featured in the main scientific magazine, one of the main ones, Nature. Yeah, about how labs, and especially our labs, have been really scrambled or mean like working really hard to identify these variants and to also understand how they arise and what's the effect. And that's it. It is a picture of a patient in a South African hospital. So what we did in South Africa with initial funding from the Department of Science and Innovation, the South African Medical Research Council, is to set up a network of genomic surveillance. Yeah. And how we did that is to set up like 5 labs that across the country to sequence the virus in a very comprehensive way in South Africa. So CRISP is the organization that I led. It's leading this network. And another thing that we did is to put this genomics lab always next to a national health laboratory service. So that's the labs that serve all the health facilities. So data and samples could go very quick from diagnostic to sequencing and then feedback to clinicians, yeah. And that's what we found. We found this is a paper that's also coming this week. That's from the first wave of infection in South Africa, yeah. And then just to highlight here, we had quite a severe first wave, not close how severe is the second wave, But what we did is to start genotyping the virus very early. We even got the contact of the first infection in the country. And one thing that we learned is where the virus comes from. Most of the virus came from introduced in Africa through Europe, some of them from North America, but the great majority from Europe. Why Europe? 80% of the international air traffic outside the continent in South Africa is through Europe. And then these introductions now showing black set up local transmission. So despite we had hundreds of introductions from outside, we end up with more than 99% of the infections in South Africa being transmitted within the country and by citizens through each other. Yeah. And that's what we found. Yeah. Again, we start that very early in February. That's the data. And here's the proportion of the genomes. When I have white, it's not that I don't have data, it's the opposite. Normally I have 20, 30 lineages there. So just for simplicity, I'm showing in white. So what we had normally, South Africa have 20, 30, 40 lineages circulating. Around May, we start creating what you call our own lineages, yeah, which they get a number in the classification, and they are mostly created through super spreading events such as hospital outbreaks or convention center outbreaks. They follow in the population. And these lineages, they were circulating with small prevalence, 10, 20%. Yeah? But then what we found after a tip from some doctors in the Nelson Mandela Bay, it is a new lineage or new variant, which we number 501YV2. And this variant appear, we first samples that we get was in mid of November. We went back to the freezers to see when it start appearing. And as it appears, start completely dominating and displacing all the other lineages, yeah? And if I show data that came today from our lab, now is in 99% of our virus. It's only one virus circulating in the country, and that is this lineage. And that's one of the main data that could show us that this variant is much more transmissible. Not only it completely dominates, but it is traveling very, very, very fast into many different areas. Yeah. So here's just a map.
Tulio, do you mind going back to that last slide? Because I think that slide is so important that you produce for us to explain to the lay audience. So the yellow bar that started in October, if you look by September, there basically wasn't any of that yellow bar, which is the variant that you now discovered, called, I call that your 484 variant. And this is all just South Africa. So by what you're saying, by November, that yellow bar had taken over the proportion of genomes of infections, 90% that yellow bar speaks to the rapidity of the infection transmissibility and what I call genomic surveillance, really credit to your strength of genomic surveillance and the importance of pandemic surveillance and pandemic awareness, very much like battlefield awareness. You need to know what enemy you're fighting.
Yes, thank you, Patrick. And of course, that also the way that we did is to do something that sometimes people think that's boring, but it is repetitive. Every week, get hundreds of samples from dozens and dozens of clinics and do the analysis. Yeah, and that sometimes is what's not the exciting part on science. But if you do that very repetitive and consistent, you can see change very quick. Yeah. So if I just go to the next slide, because I also find that one quite exciting. Yeah. What we can do here, and that's these lines. Yeah. And you're gonna see the different shades. The shades we can recreate the travel of this virus. Yeah. So we can identify where it started in the Nelson Mandela Bay, which was called Port Elizabeth. Yeah. Around like not only was first detected in September, but all these multiple introductions to next towns, yeah, and then which one of them quite early in blue, it go to this area in South Africa, very touristic area, the Garden Route, fantastic, beautiful place, and that it also serves as an amplification event because lots of people travel to this area to introduce the virus much further away, yeah. And South Africa is quite a big country. To drive from KwaZulu-Natal to Cape Town is like 2,500 kilometers. It's quite far. Not as big as the US, or what would be like 1.7 thousand miles, yeah, but it's quite far. And the speed of the diffusion of this virus I'm going to show you in the next slide, yeah. Also the power of do that kind of analysis thoroughly, yeah. And what we did is to sample over 100 clinics, yeah. So just to make sure that what we are seeing is being replicated, and that's the kind of suggestion that I have for United States of America, to also create a network to do weekly surveillance, but also across the different states, yeah. And this is the graphic that shocked us a little bit, yeah. Here is the Nelson Mandela Bay. And the different shades in red is what we call test positivity. Yeah? And the test positivity in that area around October was getting closer to 20%. What it means, every 5 tests, 1 is positive. The World Health Organization considered a pandemic when the test positivity passed 5%. Let's just see the speed of diffusion of this virus across the coast. Yeah. So here, just moving to the weeks, and you're gonna see many of the areas become very dark red. Yeah? And this it means that all these areas, every 10 tests, more than 3 are positive. Yeah? What it means is that the size of the pandemic is multiple times higher. And we have never seen that. The speed in 10 weeks, moving to all the coast and completely dominating all the areas with very high level of infections. And let's just look at that again. Yeah. You can see moving from the center, going to the south, east, west, and the northeast, very, very quick. Yeah. And we didn't see that in the first wave. Yeah. If you go to the next slide, and here again, put in the whole country, you can see that by January, not only the same thing happened in the center of the country, but a lot in the districts that border the neighboring countries. Here would be Swaziland, Mozambique, Zimbabwe, Botswana, Namibia, Lesotho. So we have every reason to believe that this variant is now the main dominant variants in all the neighboring countries. Botswana, the last genomes that they did, 100% of them were this variant. We just received 150 samples from Mozambique, and next week we will know exactly what's the frequency of that variant. But given that all the neighboring districts have this very high unusual test positivity, we are expecting most of them to be this variant, especially because during the Christmas period, a lot of migrant workers from the country, South Africa is the powerhouse economically, would go back to the countries to spend the time with the families. Yeah. So how different is this variant? Yeah, this variant, as Patrick mentioned, that's compared with the previous 3 variants that were circulating at around 10% each in South Africa, or 15%. It has double of mutations, like 23. The other ones had like 10, 11, 12. Yeah. But more worry that not only has double of the mutation, but most of the mutations they cause amino-acid change. And that's when it changes the function of the protein. Yeah. And when we look at where that's based, yeah, as the previous lineages, they had 2 or 3 mutations in the spike protein. That's the most important protein of the virus that will bind the receptor to enter. This one has either 7 or 8 mutations. And all these mutations will cause amino-acid mutations, yeah, where all the other ones only have 1 mutation, the spike gene, the 614G, which has been shown to increase transmissibility, but this one has either 7 or 8, yeah. And then more important is to look at this mutation, the 3-dimension structure. That's something that we show the protein. So here in light green, we have what you call the receptor binding domain, which the mutation that's shown to increase transmissibility, the 501Y. And here is what we call the very bad brother, the 484K, because these mutations here in the receptor binding domain is the main area that's affected by antibodies. Yeah, This is neutralized antibodies, which is elicited during SARS-CoV-2 infection, and it's 90% of the neutralizing activities present in convalescent sera. Yeah. But we also see another cluster of mutations here that maybe we wouldn't see then if you look at linearly, such as 18, 8215, and a deletion. But this is also in another important area of the virus, the N-terminal domain. That's the other area that's affected by antibodies. Yeah? So this virus seems to have like multiple mutations and a deletion in the spike protein, exactly where you don't want it to have in the main domains for both receptor, but also the N-terminal domain. Yeah. So that's where we get worried when all the different groups in the world start studying these mutations. Yeah. In this graphic here, I have it sounds complicated, but it is not. If it's dark red, it means that there is resistance to the different antibodies, the antibody names on the top. And here is the position of the mutations. Yeah. So you're going to see this big cluster of red around the 484, especially the 484K, because this is the mutation that showed to confer the broader cross-resistance to several monoclonal antibodies and even combinations of them. So here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And here we see 6 of them with completely completely resistance, and the remaining ones with kind of high level resistance above 50%. Yeah, and that's the number of different papers that highlight that. Another crucial experiment comes from the group in Italy that they grow this virus in the lab, and they keep putting convalescent plasma, and around 11 weeks or day 80, it developed the E84K, and that's a massive decrease in neutralization. It's followed by insertion and basic. This now it's a completely immune escape mutation. So this E484K led to a 4-fold decrease in neutralizing activity, and this with 19 different convalescent plasma. So the virus in the bottom, here just to notice that's exactly the opposite. White, it's low neutralizing activity. And as you see, most of the antibodies do not seem to be working. That was the crucial results that came last week, also from Bloom Lab, from the Fred Hunch, which what he do, he map all the mutations in the receptor binding domain, so they are here. And what it showed that the mutation that they have the highest escape across the sera, this means that the one that reduced the bind activity is the 484, So it caused the largest drop in neutralization. It's easy to understand because this is a position that's highly exposed. So once it changes, it gets less affected by antibodies. So again, all this evidence is just building in the last week, of this mutation being very important. But we want to highlight that today was another preprint that came about the N-terminal domain, showing that a lot of the mutations that they have in the 501V2 and also in the V3, the one that's found in Brazil, also have the 18F, the 484K, and the 501Y. They seem to also be associated with escape mutations. So this true, and we have a deletion just before here, also with monoclonal antibodies. I know that everyone is just looking at the receptor binding domain, which probably are very important, the most important region, but would like to highlight that there is also this other region that have cluster of mutations that also support the being escape from antibodies. Yeah.
So what you were showing there, if I may, on that, because this is so important for even the scientific community, the regulatory authorities, the NIH, to understand that when you get this thing called the NTD, then the N-terminal domain of the spike protein, it's not the direct interface between the RBD and the ACE2. Yet, when you mutate here, you still would think you will see emergence then of escape from the monoclonal antibodies, meaning that these mutations outside of these neutralizing antibody assays are as important in terms of getting resistance. Is that what you're saying here, Tulio?
We wouldn't know if it is as important, but what we think that they are also important, yeah. And just to highlight, should not forget about them, because this NTD area, it also seems to have strong signs to be escape mutants from antibodies, yeah.
Which speaks to the fact that one needs to then find even monoclonal antibodies to the N-terminal domain to see if that would help.
Yes.
Okay, thank you.
So this is the kind of viral cells and the lung cells that are there happily circulating, yeah, and being replicating in the lab, yeah. So see what's happened when these are infected with SARS-CoV-2. When they are infected with SARS-CoV-2, they are circulating, the virus starts infecting them. They start doing what Patrick called very similar to cancer. They start the cell-to-cell spread, and then they start killing the cells very quick. And that's exactly what's happened in the lungs of people. That once the virus circulates there for some time, especially first as cell-free virus, but then when start infecting many cells, they start transmitting to each other and start having massive number of cell deaths, that big organelles that are being created. So once this virus is grown in the lab and you can infect them with the virus, then what you can do, you can use the convalescent plasma from previous infections and see how that will neutralize. And that's what we did here in South Africa. I'm just trying to ground you. I know that many people find it difficult to understand phylogenetic trees, but here's what you call a tip-dated tree. That's the dates where the virus were isolated. So the first thing to see is the big cluster, the 501v2. That's basic, and that's all the previous virus circulating in South Africa. And when this one appear, almost the other ones cease to exist in very small numbers, yeah? And here, what I'm marking here in orange is the virus that we give to Alex Siegel, which he grow in the lab, and this virus has all these mutations in the receptor binding domain. And here, with also another virus that he has collected from the first wave back in July, yeah, which is also growing in the lab. So he takes these three different viruses growing in the lab, and then he takes the convalescent plasma from this one, that one, that one, that one, that one, that one, and each other. And then he exposed to each other to see how they neutralize. So that's how the experiments work, yeah? Basic, all these individuals that you have the virus, you also have like blood from them, you know how they respond to the first infection, and you see how the plasma will neutralize this virus. Yeah? And I'm not gonna show you all the results, I'm just gonna give you a peek on that. So that's how it looks. There you have a focal point. Here is an uninfected cell, so you can see no infection. Here's the virus from the first wave. That's when you just leave to circulate. It's creating all these foci of infection. And then we start titrating and add different convalescent plasma. And here is at low concentration. And then we start increasing the concentration of that. And then what you can see, that start killing most of the virus infection, and you have very little that's alive. But that's what's happened with the 501v2 with all these mutations. Here is without any convalescent plasma. And as you keep adding more convalescent plasma, you do see some neutralization, but much, much less than you expect. Yeah. And that's what we're going to be publishing next week, showing very limited, very little neutralization from convalescent plasma on this new virus, which has all the characteristic of being a escape antibody mutant virus, which is much less neutralized. But again, as I mentioned in the previous slides, this building another 5 or 6 big line of evidence suggesting ours that these different mutations, especially the 484K, may allow the virus escape from antibody. Yeah. So just to conclude, yeah, genomic surveillance is a critical component of the public health response and should be scaled in most of the countries in the world. And we think that we can exemplify very well by the early detection and tracking of this new 501Y V2 variant. Yeah. We have detected a new lineage with mutation at key sites in spike glycoprotein. This mutation associated with decreased antibody response in many different experiments from our groups and many international groups in the world. From the protein structure, from the pseudovirus, from live virus, all the evidence in point of the same direction of decreased antibody response. Yeah. We crucially and very quick need to determine the clinical significance, such as the risk of reinfection and the impact of vaccines. Yeah. Just to finish, a lot of the international media will say that I'm the virus hunter and that I have done that alone. But in reality, this is the work of dozens of, if not hundreds of scientists in a very strong, coordinated, collaborative network for genomic surveillance involving all of the top universities in the country and research institutes and funded by the national foundations, both the Department of Science Innovation and the South African Medical Research Council. Yeah. Thank you. And it's a great pleasure to talk to the LA Times and to you, Patrick. Thank you.
Thank you for your time. I really appreciate this conversation, and I look forward to having much more conversations like this. I am sure you'll be sought after, but I really thank you for your time to give us at the LA Times this opportunity.
Yeah, thank you. It's a pleasure. And the serious media has a very important role on this pandemic and to bring science to the forefront of all the public. So thank you, Patrick, and thank you for your editorial team.
Well, I look forward to coming back home to work on this project with you. So keep well, keep safe, and keep working. The scientists of the world are entering into an amazing phase of incredible collaboration and collegiality. We have tools in our hands we've never had before. The genomics analysis that Tulio and his team have done, the capabilities of actually now building next generation vaccines as rapidly as been done with the mRNA vaccines, and the idea now of our insight, our understanding that what we call immune pressure and viral evolution is here, but we do have tools to fight that. And the tools to fight that is the T cells. You and I have T cells. We have NK cells. We have macrophages. So your immune system is available to fight that. We will not give up. The opportunity for this virus to take over this planet is not at hand. The human race coming together has a way of overcoming, and we will overcome. I am personally incredibly excited that we will actually now be launching the trials of our T-cell vaccine in South Africa, where we're going to take on this mutation and go into the eye of this storm and hurricane and take on this mutation. So with that, I wanted to thank you for listening to this third series of the science behind coronavirus mutations, and look forward to presenting to you more information as we proceed further in our series on the second opinion. Thank you for being with us.